So far in this presentation, we've created the argument of why we need research for both our own practice and also the creation of guidelines. We've also looked at the philosophical underpinnings of quantitative and qualitative research. Now in this presentation, we're going to move on to look at quantitative research in a little bit more detail. So far, we've learned that the standpoint of quantitative research is that everything can be measured using numbers and scales. The underpinning thing about quantitative research is control of variables and manipulation of variables. And in this presentation, we're going to look at the top three: independent, dependent, and confounding variables in quantitative methodologies. In quantitative research, variables come in three types, and these are things that we're either looking to manipulate or to observe. Often the one that's manipulated is the independent variable, and you can think of this that is independent to everything else. And this is often the treatment or predicted relationship. And this can be a drug that's given, a placebo that's given, or two different cohorts of patients or practitioners that have been observed at any one time. From this, what we're looking to discover is the dependent variable, and this is the outcome. And you can think of this that the dependent variable is the result of the interactions between the independent variables. So let's take a look at a research question, and we'll understand this a little bit more easily. So here we've got a research question. Experienced paramedics are less likely to express positive comments about extended roles than less experienced paramedics. And this is an actual research question from the literature. We can pick out the treatment or predicted relationships as the experienced or less experienced paramedics. And these are either ones that we're looking to observe, or we could manipulate to have them in one group or the other. The outcome is the positive comments. And we can see here it's quite easy to pick out the independent and dependent variable. So let's take a look at another research question to make sure we understand independent and dependent variables. So we've got one here: patients who receive a copy of a leaflet about self-care are more likely to stay at home than those who do not. And let's try and pick out where we think the independent and dependent variable would be. First of all, let's have a look at the independent variables. We can see that what we could potentially manipulate, the treatment or predicted relationship, would be those patients who receive a copy of a leaflet or those who do not. And we can see that the outcome of receiving that leaflet or not would be whether the patients are more likely to stay at home. And this is the crux of quantitative research, that we manipulate the independent variable, or sometimes don't manipulate, to produce the outcome, the dependent variable. So let's take a look at this independent variable a little bit more closely so we know that we understand it. We've already learned that it is presumed to affect the dependent variable. But I've alluded to that we can manipulate this or not manipulate this. And this goes to say if the research is experimental or if it is non-experimental. In experimental research, the independent variable, the treatment, is manipulated. And we can see this in the COVID trials, for example, where one set of patients is given a vaccine, one set of patients is given a placebo, and by that we are manipulating the independent variable to see what the effect is on the dependent variable, which would then be if the symptoms are reduced and if patients are admitted to ICU or not. However, we could make that into a non-experimental study. So what we could actually do is leave it up to chance, and we could actually not manipulate who gets the vaccine and who does not, and then see what the effect on the dependent variable is. So randomized controlled trials or quantitative research can be experimental if things are manipulated in the independent variables, or non-experimental if they're just left to occur naturally. Let's take a look at our research question and decide how we could actually make it an experimental or non-experimental study. We've got the same research question here about patients receiving a leaflet or not, and deciding if this is going to make them more likely to stay at home. So we'll say you've got a patient with a low-grade fever and that you don't want them to go into hospital, but you want them to self-manage with paracetamol, for example. You could give them a leaflet about paracetamol and self-care, and that would hopefully not impact them and make them go into hospital. So we could make this experimental by taking a group of patients, say 50 patients. We could give 25 the leaflet, we could give 25 no information, and see if they stay at home or not. So we've made that experimental by manipulating that independent variable. However, we could leave it up to chance and leave it up to the paramedics to decide if they give a leaflet or not. We would then follow up the patients that had the leaflet, follow up the patients that didn't, and then see the effect on the dependent variable to see if they've gone into hospital. So quantitative research is all about manipulating what we do to produce that dependent variable or not, but it's also about potentially letting things occur naturally and seeing what the effect is on the dependent variable. So let's take a look at this dependent variable and make sure we understand the significance of it. We've already learned that the consequence of the dependent variable is what we do to the independent variable. Do we manipulate it or do we not? i.e., do we give a patient a leaflet or do we not? This can change depending on the independent variable. The important thing in quantitative research is that we do not manipulate this dependent variable. This must remain completely separate to everything else within the research, or we would not know if the action of what we'd done or what we'd observed had actually been produced in the dependent variable. So for example, let's take a look at our research question again of giving patients a leaflet in order to stay at home and self-care, or do not receive a leaflet, and if that would impact them going in. We've already established that the independent variables are whether we give a patient a leaflet or whether we don't. The dependent variable of them more likely to stay at home is something that we can't interfere with. We would have to give the patient the leaflet and the information and let them make their own decision. What we couldn't do is give them the leaflet and then advise them to go into hospital, because what we have done there is created bias and introduced error into the study by telling the patient to go into hospital. We have therefore interfered with the dependent variable. So a key point is to make sure in the papers that you have a look at in quantitative research that the result, the dependent variable, the outcome, has not been messed with in any way whatsoever. So you might be thinking, well, that's great, Zach, but can we really control every element of a trial based around just independent and dependent variables? And the answer to your question is absolutely not. We do get these other things called confounding variables, and what they are is variables that may influence the relationship. And what these do is compete with the independent variable, that giving of a leaflet, and potentially produce a dependent variable that was not caused by those independent variables. So let's take a look at an example of this in our research question of a patient who receives a leaflet. We've got our two independent variables, the leaflet's either been given or not, and we're hoping to see that that will have an effect on whether they're more likely to stay at home or not. What we would hope is that patients who are given the extra information about their condition or self-care or management at home would then not go into the hospital and therefore are more likely to stay at home, the dependent variable. But because we're dealing with humans and not situations created in a laboratory, we have these things called confounding variables. So let's take a look at examples. We might have a set of patients who are given the leaflet but don't actually read it and then go into the hospital anyway or don't stay at home. So we're not sure that that's a direct action of the leaflet or whether it's their decision not to read it and stay at home anyway. We might actually have a patient who's given a leaflet who is actually afraid of hospitals and therefore no matter what the leaflet said, they would stay at home anyway. Conversely, we might have a patient who doesn't mind going into hospitals, who is possibly a bit of a hypochondriac or a frequent attender to the emergency services and emergency department. Therefore, the information that you give them is really going to have little impact, and it would still mean that they go into the hospital. You might also have patients who have maybe not received the leaflet as part of the trial, or have maybe lost the leaflet and therefore not read it. Again, this is a confounding variable that could produce that dependent variable, that outcome. You may also have a patient group who are wrongly influenced by the leaflet as well, and who, given the information, may decide to go in or may decide to stay at home as well. So there's lots of confounding variables that could happen within this study that we need to account for in the reporting of. And what we've got to be aware of when we read research is that, especially if it's an experimental research, is it really the independent variable that has had an impact on the dependent variable, or are there multiple confounding variables that have produced the dependent variable? Therefore, it's unreliable if the independent variable actually influenced the outcome.